everyone for joining. Thanks everyone for joining us today. I can see lots of students here. I wish I can say each of your name. So maybe later on when I ask questions, without any further delay, I would start sharing my screen and I will start having uh, my presentation. So I will just minimize this and make it bigger. Hi everyone, my name is Noeen and today I will be talking about mRNA derived vaccines. Before I start this lecture, I would like to give acknowledgement to following articles. This lecture is prepared according to the reported data, which is published in literature in the form of following review articles. All these review articles are pretty new and they are really well written. If any of you would like to have a copy of these review articles, please let me know if your library has not access to these journals, and I can definitely send you by email. They are really worth reading. The outline of today's lecture would be about outline, which includes my journey, vaccines, in which I will cover like types and the comparison. Then we will move on towards mRNA-based vaccines, which includes the timeline development, methodology, and then the manufacturing, which is again the development. Under development, we will learn today about antigen designs, vector designs, delivery system, which is required, and manufacturing. Then we will have a brief overview and we will go towards COVID-19 vaccines, their status, and Merck's pill, which is the most recent development for which they have already gotten some permissions for a trial. And then we will have some key takeaways or the questions. So I would like to start with my career. I started my career as a scientist, like after my undergrad degree as a research assistant in Germany in radiopharmacy department, University Hospital Tübingen. And in 2008 to 2011, I pursued my PhD in radiopharmacy, which is basically nuclear or radiochemistry in nuclear medicine. And then I started working as a research scientist in 2011. In 2012, I went to US for a postdoc for 1 year. Then I moved back to Germany in 2013 as a senior researcher and faculty for CGMP production of radio tracers for patients. In 2015, I moved to New York, Department of Radiology, New York School of Medicine, New York University, US, to work as a research associate. There I worked with mainly with PET CT scanners for diagnostic purposes for cancer, oncology, and neurodegeneration diseases like Alzheimer's and Parkinson's, as well as I worked with SPECT scanners. Then I moved to Canada in 2018 to work in TRIUMF, which is the Particle Accelerator Center, and in BC Cancer in Molecular Oncology Lab to work as a postdoc fellow in research and development. And also in 2019, I worked for 6 months as a commercialization intern with TRIUMF Innovations to bring the innovations to the market as a business strategist. And I worked there until 2020 September, and after that I joined some organizations as a volunteer, and I worked with, and then I got in 2021 board position as an executive director of public affairs in Giant and director of communications and events with Quist. And I recently, since August, I also am a founder and CEO of Scientudio. And also I am an incoming research scientist for in USA, for California. I'm just waiting for some of my documents to complete. Before we start this our presentation or lecture today, I have just one question: that does anyone know what from which words the terms vaccine and vaccinations were derived? If you know, kindly unmute yourself and let me know. And if you don't know, then definitely I will move on with the answer, and I will continue my presentation. Anyone? Okay, so the terms vaccines and vaccination. What? Sorry?
Immunization.
Okay, got it. So anyone else? Okay, so the terms vaccine and vaccinations are actually derived from a Latin word for cow vaccines. Edward Jenner, he was the doctor, British doctor in 1796. He referred to cowpox as variole vaccine, which means smallpox of the cow. So this is how the term vaccinations or vaccines came from. So now we will move on towards our types of vaccines. I have now based my lecture on COVID-19 virus, which is everyone knows due to the pandemic. Um, Types of vaccines can be derived into two types of the main platform. One is the classical platform, one is the next generation platform. In classical platform, there are different varieties. One is some are the virus based, such as whole inactivated, live attenuated viruses, virus like particles, and protein subunits. Whereas the next generation platforms include the viral vector, DNA, RNA, and antigen presenting cells. I will talk about little bit details in my next slides. But before we go, I would like to mention some disadvantages of the classical platforms. In the case of COVID-19, for example, if we would like to move on with classical platforms, large quantities of viruses would need to be grown under biosafety level 3 conditions for a whole inactivated virus. For this, extensive safety testing is required to ensure that live attenuated viruses, for example, are safe and they do not easily revert to the wild type. So this is the main disadvantage which is related to the biosafety. Whereas in case of the next generation platforms, the main advantage is that they can be easily developed based on the sequence information alone. If viral protein important to provide the protein protection from infection or disease and vaccine antigen is known, the availability of coding sequences for this viral protein suffices to start the vaccine development rather than having to depend on the availability to culture the virus. This makes this platform highly adaptable and speeds up the vaccine development considerably. I will talk about this DNA and RNA vaccines in my next slides, but I would like to mention a little bit small information about antigen presenting cells here. Antigen presenting cells are essential component in the immune system's response to a vaccine. Loading antigen presenting cells with peptides that would otherwise be produced by vaccination bypasses the first step after vaccinations. Traditionally, dendritic cells are harvested from the individual, then expanded and manipulated to present the desired antigen and infused back into the same individual. This is cost prohibitive and too time consuming for a vaccine to be developed on a large scale. So this is the main disadvantage of having the anti-presenting cells based next generation platform of vaccines. Now I will start talking briefly about our some details based on COVID-19 vaccines. This is the first platform or the convention platform category is inactivated vaccines. These contain killed COVID-19 virus, and actually this virus is recognized by the immune system to trigger a response without causing illness. This response builds immune memory so your body can fight with the COVID virus in the future. This virus based on COVID-19 inactivated virus is no longer, which is no longer infectious, since whole inactivated virus cannot replicate. So adjuvants are required to stimulate the immune system together with the vaccines. The considerations are the same thing, that the adjuvants are actually required to boost your immune system. Examples in human use are influenza vaccine. Approved elsewhere is the Sinovac, Sinopharm, and Bharat Biotech. In clinical trials, Shifa Pharm, Chinese Academy of the Medical Sciences. Now let's move towards the attenuated viruses. Another virus-based vaccines, live attenuated vaccines, are classically generated by passaging the cell culture until it loses its pathogenic properties and causes only a mild infection on injection. So in other words, these are called as the weakened viruses, which is recognized by the immune system to trigger a response without causing the illness. This response builds immune memory so your body can fight off the virus in the future. A well-known approach which requires time and extensive testing, it resembles the natural infection. Examples are oral polio vaccines, such as the Cordagenics. Now we will move towards our protein-based uh, vaccines. So protein-based vaccines can consist of a protein purified from a virus or virus-infected cells, recombinant protein or virus-like particles. Virus-like particles consist of structural proteins necessary to form a virus particle, but lack the viral genome and non-structural proteins. Protein-based vaccines require also the addition of adjuvants to induce a strong immune response, which can also come under our considerations. These viruses can be the whole proteins, protein fragments, or protein molecules packed into the nanoparticles. And the examples for human use are hepatitis B vaccine. In clinical trials are the Novavax and Sanofi GSK. Maybe for hepatitis B vaccines, you know that it is also related to cancer or oncology, liver cancer. The next type of vaccines which are virus based are the viral vectors. This is also the type of the next generation virus vaccines. These consist of a recombinant virus, often attenuated to reduce its pathogenicity, in which genes encoding viral antigen have been cloned using recombinant DNA techniques. Vector vaccines can either be replicating or non-replicating. Replicating vector vaccines infect cells in which the vector antigen is produced, as well as more infectious viral vectors able to infect new cells that will then also able to produce the vaccine antigen. Non-replicating vector virus vaccines, on the other hand, initially enter the cells and produce the vaccine antigen, but no new virus particles are formed. Because viral vector vaccines result in endogenous antigen production, both humoral and cellular immune responses are stimulated. Um, if you see the consideration, it may need to be stored at specifically low temperatures. The one of the example is the Ebola vaccines approved for COVID-19 is the AstraZeneca from Oxford. The other vaccines approved are the Janssen's, CanSino, and Gamaleya. Let's go towards now the genetic vaccines, which are mRNA and DNA-based vaccine, which is our today's topic. So these nucleic acid-based vaccines consist of the DNA and mRNA and can be adapted quickly when new viruses emerge, which is why they were among the first COVID-19 vaccines to enter the clinical trials. DNA vaccines consist of synthetic DNA construct encoding the encoding the virus and antigen. For efficient uptake of the construct into the cells, injection needs to be followed by the electroporation in case of DNA vaccines. After uptake into the cells, the vaccine antigen is expressed from DNA construct. Whereas mRNA-based vaccines work on the same principle as DNA vaccines, except that the first step, nuclear translocation of the DNA construct and transcription into the mRNA, are bypassed. Self-replicating RNA vaccines are likely to induce protective immunity at a very lower dose because more vaccine antigen is expressed per cell. However, since mRNA vaccines are not very stable, these constructs include modified nucleosides to prevent degradation. A carrier molecule is also necessary to enable the entry of mRNA into the cells. Lipid nanoparticles are most commonly used for this purpose. So that's why their consideration is the low cost and fast develop, but also they need to be stored at the extremely low, specifically low temperatures. And the approved COVID-19 vaccines falls in this category are the Pfizer and Moderna. In clinical trials are the CureVac and in Innovio Pharmaceuticals. Now let's talk about mRNA-based vaccines, their timeline. If you see, it didn't happen just during the year of pandemic, just in 1 year. This research actually, it started in 1961. You can see with the discovery of mRNA isolation, and it continued over the decades. If you see, it is almost the work of more than 20 and 30 decades. And in 2017, when it was first mRNA based vaccine was developed for influenza, and then in 2020 they started clinical trials for influenza vaccine, and in 2021 and in 2020 they also used the same methodology and approach to prepare these such vaccines for COVID-19. Now let's go. I would like here, it's a very simple and small video for you to watch. I would like to share it with you. It is from Harvard YouTube channel. You can go and follow this link and watch it there too later on. So
researchers have found that the COVID-19 virus begins the process of creating copies by attaching its spikes to a host cell. Those spikes offer a target for vaccines. With the right preparations, our immune system can utilize this vulnerability. A vaccine is used to arm our immune system. It contains messenger RNA, which can direct cells to make specific proteins. The messenger RNA tells our cells to produce COVID spikes. These harmless spikes give our immune system the information needed to quickly detect a coronavirus infection and produce the appropriate antibodies. The new antibodies respond once COVID has entered the body. They attach themselves to the spikes on the COVID virus, rendering the spikes useless, protecting our cells and keeping the virus from reproducing. mRNA vaccines have been studied rigorously and are a safe and effective way to defend ourselves against COVID-19 infections.
okay, thanks everyone. So maybe you have watched, this is a very simple 1 minute video how mRNA vaccines work, and you can even share with your contacts and your family and friends who would like to know in a very simple and lay language. So let's go towards our next slides, which is now we will move on towards our methodology, and we will start first with our mRNA based vaccines in first one. So basically, methods to encapsulate mRNA have also been explored to increase the stability and immunogenicity of the RNA vaccines, as has been used with exosome encapsulated RNA and RNA transfected dendritic cells. When fully optimized, RNA vaccines have an immunogenic advantage over DNA vaccines due to the presence of multiple cellular pathways that activate the innate immunity in response to foreign RNA, such as the toll-like receptors. So let's see into the methodology of these vaccines. Okay, I go here, like just give me 1 second, please. Okay, so the first type is the RNA engineering. This is done actually by here the efficacy can be increased by several factors, such as ensuring the mRNA purity, adding the 5-cap COSAC. You can see here adding 5-cap and COSAC sequences, and 3 poly-A sequence to mimic the structure of endogenous mRNA to avoid the detection by innate immune sensors. Also by decreasing the detection of by decreasing the detection by the codon optimization, also introduction by intramuscular and intradermal injection to reduce the RNA degradation, and by generating the thermostable mRNA, which is the second type. Both of these are actually widely used in the drug design. In case of the thermostable RNA, it actually involves freeze-drying RNA and incubation with various biomolecules to increase RNA stability even at higher temperatures. Now we move on towards our dendritic cells-based vaccines, or the methodology in which RNA is actually transfected into dendritic cells in vitro, which can then activate CD4 T cells and stimulates antibody production in the vivo. These methods to encapsulate have been explored to increase the stability also, and it involves the it involves actually the exosomes encapsulated RNA and RNA transfected dendritic cells. These also help to increase the immunogenic or response of the RNA based vaccines, and due to the presence of multiple cellular pathways that activate the innate immunity, such as the toll receptors, TLRS, and RIG1 like receptors. These are mostly explored for use as in cancer. The other one are the self-replicating RNAs. So vaccines can be self-replicating or self-amplifying RNA molecules that are normally based on positive strand RNA viruses, of which the structural genes are replaced by the antigens. Both trials have similar safety profiles, with most patients experiencing mild to moderate reactions to administration and a few patients experiencing little bit severe reactions. Both vaccines are demonstrated immunogenicity through neutralizing the antibody level, though antibody levels with rabies vaccines were more highly dependent on dose and rate, with needle-free intradermal dose able to sustain neutralizing antibody in half of the number of vaccinated individuals 1 year after the injection, but not with those receiving intramuscular or intradermal injections. Both have been also both type of the vaccines are already also in preclinical and also have been based for main basis for the mRNA vaccines for the COVID-19. Now let's go towards the development, how these vaccines are basically developed. The first pillar is actually the antigen design, and let's talk about a little bit more about antigen design. So since there are actually 4 main elements to be considered or to look for it. First are like the viral glycoproteins. So self-amplifying mRNA vaccines are actually encoding the viral glycoproteins, and this is the most dominant application. Though these recently been expanded to include the bacterial infections also, such as chlamydia, trachomitis, group A and group B, streptococci, parasites, and cancers such as colon carcinoma and melanoma. All the clinical trials currently underway for the self-amplifying mRNA using the viral glycoproteins as their targets. These glycoproteins are present in the spike proteins. Most of the COVID-19 vaccines, they use the spike protein glycoproteins as their targets. The another type is the bacterial antigens. So these self-amplifying mRNA vaccines against the bacterial antigens have also been investigated, although are limited to protein targets as opposed to polysaccharides and non-protein surface makers. Self-amplifying RNA formulations all exhibited antigen-specific humoral and cellular immunity against MOM, although a challenge study was not included in the part of the study which was reported in the literature. These studies show that it is possible to use these type of the vaccines against bacterial pathogens as a means of disease prevention. The other type that you have to consider during your making your construct for mRNA self-amplifying vaccines is about the parasitic antigens that also you can take into considerations. It has been to date applied to the 2 types. One is the Toxoplasma gondii and the Plasmodium. There are so many studies reported already in the literature, and they demonstrated that self-amplifying RNA platform can also be used to prevent the parasitic infections. The last actually and the most important and the most novel approach includes encoding the monoclonal antibodies for passive vaccination. While it is possible to incorporate the relatively large number of multiple antigens into your self-amplifying RNA vaccines construct, the DNA construct does have the size limitation. So it may be advantageous to use these monoclonal antibodies for the passive vaccinations, to use separate saRNA constructs to encode the multiple antigens if necessary. Here, I would like to have a questions for you for as a tester. I think we already discussed about it. Which vaccines other than human papillomavirus vaccine or HPV prevents a form of cancer? It is also very much dependent on the antigen, actually, antigen design. Anyone knows about it? Actually, it already came before. I didn't notice it. So if any one of you know, just unmute yourself and let me know. Anyone?
Can we say the hepatitis vaccine against liver cancer?
Yes, it is hepatitis B. So before I move on, I would like to talk about the disadvantage of these monoclonal antibodies for the passive vaccinations. As opposed to the active vaccinations, these type of the vaccinations actually provides more immediate protection against a pathogen, but they are very expensive to produce and difficult to administer. So that's the reason that they are not being in use, or they are still in the preclinical trials. So it still needs a long time for this type of strategy to be in a practical application. So let's go toward, this is anywhere used against the cancer or rare or inherited diseases still in our preclinical trial or studies. Next is about the vector design that you have to take into the consideration. Let's talk about the vector design. If you see in this slide, there are two types of the vaccinations: conventional, which are the non-amplifying messenger RNA, and the self-amplifying messenger RNA. In self-amplifying messenger RNA, if you see what's the difference with the conventional conventional and non-amplifying mRNA, the difference is the difference is that self-amplifying RNA vaccines have considerably larger than 9 to 12 kb than non-amplifying mRNA constructs. They contain basic elements of mRNA: a cap, 5 UTR, and 3 UTR poly-A allyl. This is the tail of the variable, as you can see on the right hand corner, and non-structural proteins, which you can see here from 1 to 4, and a subgenomic promoter, which you can see here between the antigen code and the non-structural proteins. The genes in the viral genome that are normally a downstream of the subgenomic promoter and encode the viral structural proteins are replaced by the gene encoding the vaccine's antigen. The deletion of the viral structural proteins renders the mRNA incapable of producing an infectious virus. After delivery into the cytosol of the cell, the released mRNA is traditionally competent and engages with the host cell ribosomes to produce 4 functional components of RNA-dependent RNA polymerase or viral genome replications apparatus, which are the non-structural proteins such as 1, 2, 3, and 4. Now there is another type of the mRNA-based vaccines, which is the trans-amplifying mRNA-based vaccines. So if we know that it is very well known that self-amplifying mRNA vaccines actually activates the type 1 interferon response through both endosomal sensing via toll-like receptors. While this is advantageous for enhancing the immunogenicity of the sRNA vaccines, this IFN activation also known to lead to inhibit the translation and degradation of the cellular mRNA, which may hinder the potency of the vaccines. To counter these, there is a next generation design, which is called as a trans-amplifying mRNA. This trans-amplifying mRNA, if you see here, uses 2 types of the transcripts to achieve a similar effect of the to the self-amplifying RNA. A conventional mRNA encoding, which you can see here, and as well as including the coding of the self-amplifying RNAs. These actually help in, these are actually next generation designs, and they also help the incorporation of interferon inhibiting factors directly into the vector backbones. A conventional mRNA encoding is flanked by the 5 and 3 UTR, as you can see here. It is also present here, but here it is replaced by 5 CSE and 3 CSE, which is the conserved sequence elements here. So that's the difference, if you can see in these pictures. So they just increase the immunogenicity, but in the same time, they decrease the activation of IFN. The next is the delivery system, which delivery system you are using, basically. So for delivery of the self-amplifying mRNA, there are different components or different targets that you can use. One is the lipid-based delivery, polymer-based delivery, peptide-based delivery, virus-like replicon, cationic nanoemulsions, naked mRNAs, and dendritic cell-based mRNA vaccines. So for lipid-based delivery, let's talk about this first. So lipid nanoparticles have been widely used as carriers to deliver mRNAs. These lipid nanoparticles are mature negatively charged nucleic acid delivery platforms characterized as ionizable aminolipids, polyethylene glycols, phospholipids, and cholesterols. To be more specific, these lipid nanoparticles have two advantages as RNA vaccines vectors. On one hand, LNPs defend the mRNA from degradation by enzymes from endosome. The characteristics it guarantees the high encapsulation and efficiency. On the other hand, they have good biocompatibility through a series of biological processes to deliver mRNA for expressions. Now let's talk about the polymeric based delivery. These polymeric materials are less clinically investigated as ionizable lipids too. However, they coat mRNA without suffering from degradation and promote the protein expression. However, there are disadvantages. The disadvantages of polymeric materials are polydispersity and the clearance of the large molecules. To improve these therapeutic effects, many other components have been added in these constructs, such as the lipid chains, expand branch structures, and construct biodegradation promoting domains in order to improve the clearances. This classification of the polymers contains the cationic polymer and anionic polymer. Although the cationic polymers are the dominant materials, anionic are also sometimes used to deliver. Since the negatively charged mRNA is uneasy to be delivered by the anionic polymers, the addition of cationic lipid in the PGLA complex can assist in establishing an efficient RNA encapsulating system. Collectively, polymer-based delivery system is a promising platform for mRNA delivering efficacy. However, there are still investigations going on. Now let's move on towards our peptide-based delivery system. So due to the electrostatic interactions, you know that peptides always have charged, positive and negative charged ends. The negatively charged mRNA is delivered easily by cationic peptide. The amount of loaded mRNA is positively correlated with N and P, which is the negative and positive ends or the ratios of the peptide. Additionally, an increased ratio of the charged amino phosphate groups can increase the zeta potential and minimize the particle size, increasing the encapsulated efficiency. Now let's move towards our virus-like replicon particles, which can encapsulate antigen encoding saRNA for delivering into the cytosol, which is like a virus infecting manner. The viral structure proteins are synthesized in vitro, followed by encapsulated designated antigen encoding saRNA. Some attenuated viruses maintain the ability of self-replication. Most recently, alpha virus-derived replicon RNA coding the COVID-19 spike protein has been encapsulated in lipid inorganic nanoparticles, which are also called as lines. Let's move on towards our emulsions. But before I move on to that, I just would like to mention two limitations for these virus-like replicon particles. The first limitation is that the large-scale production is not realized. The current consuming production process is limited by producing these from the cell lines. The second is that it would promote the anti-vector antibodies production, impeding the ongoing clinical trials. So now let's move on towards cationic nanoemulsions. What are these? This is actually a non-viral delivery system which potentiates the mRNA vaccines by binding to the saRNA. One of the most essential components in this is actually the cationic lipid, which is also called as DOTAP. Cationic lipids can form a pH-dependent non-sized emulsions. Based on the above many preclinical studies, these has a potential in human clinical evaluations. Let's move on towards our next type, which is the naked mRNA-based delivery system. It is different from the carrier-based mRNA vaccines. These are actually delivered directly, injecting the mRNA solution. These naked mRNA cannot cross freely through the membrane. However, it is still under several studies. This also has several advantages actually. First, that they would not be integrated into the genome. Then cytosol located ribosomes combined with the mRNA directly instead of DNA transferring from the nucleus to the cytosol. The third thing is that once mRNA reach the cytosol, the translocation process initiates immediately. This advantage determines the rapid immune responses after mRNA administration. The fourth advantage is that the final location of mRNA determines the location of protein expression. And finally, mRNA excels DNAs in decreasing the toxicity and immunogenicity. However, when it comes to the disadvantages, the vulnerability of RNA's degradation first come to our mind. However, instability of RNA in serum can be compensated by changing administration method and appropriate chemical modification. And if we think about that, then dendritic cell-based mRNA vaccines actually have advantage over it. However, the primary reason that dendritic cell-based mRNA vaccines have advantage is because it internalize, process, and present antigens to the immune cells, which generates the effective adaptive immunity very fastly or efficiently. Let's move on towards our next thing, which is basically the manufacturing, the final part. So which elements you have to take into the consideration for this? There are actually not so many publications describing the large scale manufacturing of these. However, there is a unit operations that we can follow. And here I would like to talk more about it. The first thing is the capping strategies. Which capping strategies you are going to use? So capped mRNA is actually produced enzymatically in a bioreactor, and DNA template is digested. The in vitro transcribed, or which is also called as IVT mRNA capping, can be capped either by post-transcriptional modification using capping enzymes based on the recombinant vaccinia virus, or by adding the cap analog during in vitro transcription. Enzymatic capping is more complex, but it provides the higher yields, and it also efficiency is nearly 100% efficient. And all capped structures are added in the proper orientation. Enzymatic capping is used for the large scale and laboratory productions. To increase the capping efficiency, our trimer analogs such as clean cap can also be introduced, which incorporates cap1 structures. So this is the basics. This is the first step which you need to consider when you are talking about the manufacturing of self-amplifying mRNA vaccines, and it is basically the capping strategy. Let's move on towards our purification techniques. There are different purification techniques which are used. So As you know that mRNA has a negatively charged phosphodiester backbone, and many of the purification techniques used for pDNA could potentially be adapted to the use of purification of this molecule also. And many DNA based purification techniques such as the size exclusion, reverse phase chromatography, and ion exchange chromatography, hydrophobic interactions, thiophilic adsorption chromatography, which are basically used for DNA purifications or DNA removals from different samples, can also be applied here. In addition, for routine preclinical work and in vivo immunization studies, RNA can also be precipitated. The polar nature of the negatively charged backbone of the RNA actually makes RNA highly soluble in water, and several cations, lithium chloride is which is most widely used in combination with ice cold ethanol as a co-solvent, can simply neutralize the backbone charges and decrease the solubility of mRNA, and it helps to precipitate the mRNA out of the solution. However, this technique is non-GMP and cannot be applied for the GMP productions. So then there are some other techniques which can be used in combination, which basically involve ion exchange, affinity chromatography, and size exclusion chromatography, which is basically a technique for DNA purification too. Let's move on towards another purification technique, which is the most recent one, depending on the size of mRNA, because they are in the order of 10,000 or bases or larger than 3 million Dalton. That technique is basically tangential flow filtration chromatography. So when we are going to implement it for a process which involves the GMP production, which is most of the times extremely challenging, the self-amplifying mRNA with larger size can be actually purified using strategies such as tangential flow filtration or TFF, DNA fractions, transcription enzymes, reagents, and byproducts are all removed using these chromatographic purifications for like before, which we use, followed by this TFF technique. In this during this TFF, due to the large size of these RNAs, lower molecular weight species are removed. If appropriate, molecular weight cutoff membrane is selected depending on which molecular weight is desired during the purifications. After that, this RNA can be dye-filtered into the appropriate buffer and adjusted to the required concentration. As a last step, these are then sterile filtered and stored in the bulk, ready for further downstream processing and formulations. These manufacturing or the unit processing actually helps in retaining the stability, biological potency, and functionality of RNA vaccines into the final drug construct. And whenever you are designing these, the manufacturing of self-amplifying mRNA vaccines, you always have to consider about the designs like that we talked before, that the antigen designs, your vector designs, and then of course you have to think about the delivery systems. And when you are going to synthesize it, then you have to think about the capping strategies that we talked before, and then you are thinking about more about the purification techniques which you are going to use. The next I would like to go towards my next slide is about more about like advantages, which is basically the overview, like advantages and disadvantages of these vaccines. So if I would like to divide it into the few properties. First is efficacy, which is the most important thing whenever we are thinking about the therapeutics. Then is the safety, how safe these vaccines or how safe that any drug can be. Then is the synthesis, how affordable that method can be, how easily it can be commercialized, how easily it can be adapted according to the economical conditions. And then is the delivery by the non-virus NVVs, particle systems, actually, viral vectors. So let's talk about the efficacy. If we see about the advantages of all the self-amplifying mRNAs, and including the first generation and also including the trans self-amplifying mRNAs for both, the advantages are that comparable efficacies to the traditional protein-based vaccines, high level of RNA amplification in situ. Self-amplifying RNA activity occurs in the cytoplasm, so nuclear import is not required as for DNA vaccines. Humoral and cellular responses are elicited against the expressed antigen. Protection against the infection has been demonstrated in the preclinical studies. However, related to efficacy, there are certain disadvantages. For example, prime boost administrations may be required. Little information is available on the effects of sustained high-level amplification and expression of self-amplifying RNAs. Let's move towards its safety. Viral genes for structural proteins are removed from the RNA replicon to prevent the viral assembly. Cytoplasmic mode of action, so no danger of integration into the DNA construct. Little information is available regarding the immunogenicity of the RD-RP complex and limited clinical data to date. So this is the main of the disadvantages. It is basically immunogenicity of the mRNA, basically vaccines in general. So in synthesis, the first thing is, of course, how you are going to produce it on the larger scale using the GMP in vitro transcription method. New sequences for different antigens can be synthesized easily. Flexibility to incorporate polyvalent valent or the multi-pathogen sequences. There is no disadvantage in the synthesis of these vaccines. They are fast, rapidly done, and a very speedy process. Then delivery by the non-viral vectors. Can we deliver using the non-viral vectors? Formulations are amenable to large-scale synthesis, expression and delivery site following the intramuscular, intradermal, or subcutaneous injections. However, delivery is not typically tissue-specific. Balancing the immunogenicity of the non-viral vectors and self-amplifying RNAs is very important, and it's a very crucial stuff. I would like to talk about the status of COVID-19 vaccines, just to give you a little bit brief idea of the vaccines that we have available. I am pretty sure most of you are already vaccinated. I am vaccinated. I got the Moderna. I'm not sure which vaccines you got. I will probably ask in a minute. So there are different vaccines which are available. Like RNA based vaccines are basically Pfizer, you need 2 doses, and these are the storage conditions. Moderna, which is also RNA based, 2 doses, and these are the storage conditions. Now viral vector based vaccines are AstraZeneca, Sputnik, which is Russian, Johnson & Johnson, which is the Janssen, and here is the doses information, CoronaVac, which is the Sinovac, Sinopharm, and Covaxin, and at the end, which is the protein based, is the Novavax. And this is the efficiency against the variants. So you can see different variants, which is the mutant variants, with how all of these vaccinations are so effective against all of these variants. So B means from the Brazilian mutants, actually. So let's go to my next and last. This is the most recent topic. Actually, maybe all of you have heard a lot recently on news that the Merck's pill can also be used to fight the COVID-19 vaccine. What is basically this pill? This pill is actually molnupiravir, which is actually present in the form of 2 tautomers. And these tautomers are actually resembles actually C and U. C for cysteine, U for uridine. So, and if you see here, this is the structure of this medicine. Basically, it was prepared to as an antiviral drug for oral administration to fight the influenza. I think in 2019, it inhibits the RNA-dependent and RNA polymerase metabolized into the ribonucleoside analog and resembling the cytidine and NHC triphosphatase. So during the replication, the virus enzyme incorporate NHC and triphosphate into the newly made RNA instead of using real cytidine. So this is not recognized as an error by the virus. proofreading. So when virus is proofreading, it is not recognized as an error. So virus either reads it or confuses it as a C or U because it exists in the two tautomers, which resembles the cytidine and uridine. So that's why it results in viral error catastrophe, and it is very effective then as an antiviral drug. With this slide, I would, yeah, this is very actually effective and very funny, and the simple poster that I found online. It is made by Children's Hospital of the Philadelphia. You can even download it and you can share it, and it is a very funny illustrations. And it is, I think, if you want to convey the message how these vaccines work or which are the type of the vaccines, this is very interesting form to tell even to the children that how these vaccines work. And you can download it and you can even save it as a picture, and you can pass this information on for your social media. So from this, actually, I would like to ask all of you that what are your 3 takeaways? And I would like all of you to unmute yourself and to tell me that what you have learned today from today's lecture, and what you think I should have included, or what you think is missing, and what you think is good for you. I would like any of you to unmute yourself and tell me 3 points that you learned today. Any of
I'll-
you?
I'd love to add that, or maybe to comment or ask a question, because it's commonly asked why the COVID vaccine fails with time, unlike the other. So probably you can comment on that.
Sorry, that COVID-19 vaccine can be used in addition to?
Why it's spreading its potency? Why we have to go for the booster doses every now and then?
Because most of our COVID-19, because some like because of the mutant viruses, the mutant which are mostly available, right? So the most variable constructs which are we have seen here before in our previous slides. Let me actually go back here. So you see, these vaccines are not always effective for all variants. So it is always get the boosters. First of all, the boosters will also help us to retain, like for the herd immunity, but in addition, maybe it can be also effective for various variants which are emerging in our population actually. So I think these are the main 2 reasons that I can think of, that why these boosters are important. And I am not sure, maybe in future, not only the boosters, you may be recommended to take this pill together with these vaccinations. So this is actually, I think, my stand for that.
Okay, thank you.
Any other question or anything that you learned, or anything that you think is missed from this lecture today?
Okay, thank you so much. Thank you so much, Noeen. Yeah, I think this session, I'll just take this session maybe to invite anyone with any answer. Maybe, yeah, we can take this time as a question and answer session. Anyone with a compliment, with a comment, and a question. Thank you and welcome, everyone.
Thank you so much. Hello?
Hello.
Hey, thank you so
Hey.
much, Dr. Noeen. Thank you for the presentation. I would like to ask one question for now. Could you please explain more about those dendritic cells vaccine? I think I didn't got them properly. Please.
Yeah, sure.
Thank you.
Please, I think it was my slide here. This, I think I actually talked about it here also somewhere.
It is.
No, I think it was here. Yes, just give me 1 second and I will.
Okay. I think somewhere here. Yeah, just here.
Yeah, this one, right?
Yeah.
I will actually like to give you some examples. I think I have some examples here. So maybe you can understand it in a better way. Okay, it is actually my. Yes, okay.
And also there is something else that I would like to know. I think before this slide, the one before this one, there is one that has talked about.
Yeah. So these dendritic actually vaccinations. So let me tell you, I have actually here a few examples. I will let you know. Here actually they were made actually to increase the immunogenicity and the stability of RNA vaccines, and they have been used with exosome encapsulated together with RNA and RNA transfected. You see, these are the RNA transfected dendritic cells. When fully optimized, these have the immunogenic advantages over the DNA vaccines, because of the presence of multiple cellular pathways. So because at the end you see how this is inducing the transfected cell and how it is now going here to induce your immune system to activate the innate immunity. Like you can see here is the B cell in response to the foreign RNA and through different receptors, basically receptors, for example the toll-like receptors TLRS, RIG, I1, which is actually RLRS receptors. Because of that, and here it is actually transfected into the in vitro, and then it activates the CD4 T cells and it stimulates the antibody production. And then you can see at the end of here that there are some antibodies are being produced. These type are actually mostly produced or explored for the cancers. And actually I think I have maybe a little bit more explanation about it when I was talking about these type of the vaccines to be used as uh, for delivery system, which is here. So to such like these type of the vaccines are produce very good results and very good effects from not only the upregulation of major histocompatibility complex, which is MHC actually, and molecules for combining the antigens, the co-stimulators for providing the secondary signals, and also in addition various cytokinins for T cell proliferation and formation of CTLs. We also saw it in the slide before. I also showed you like where it was activating the innate immune response with B cells, but also secretion of the chemokines for the T cell recruitment through those receptors, the toll-like receptors that I showed you before. And but it also needs some electroporation system in addition to the injections. So that's actually a double work to do. But these are actually ideal vaccines and mainly for the cancer type. That's okay.
All right, thank you.
Any other?
Yeah. Thank you so much. I think I can see someone asking from Sumra, will the tablets be taken for lifetime? I don't know which tablets is he referring to. Maybe, yeah, Noeen, you can answer these questions. Yeah.
Yeah, this tablet, I don't know. It very much depends on the viruses, their variances and mutations, right? So there are actually some most recent technologies which are still underway and they are not disclosed for vaccines, where they are taking the RNA from the core actually, and when they are taking from that, then whatever happens, because most of the times the mutations happens in the spike proteins which are on the top, on the shell of which are the membrane, right, on the surface of viruses. So because of those mutation, many vaccines are not so effective. So sometimes they do the combinational therapy. Combinational therapy would be probably vaccines and the pill together, right? But if in future there can be some vaccines which are very effective against those mutations, which are because those vaccines are based on the core mRNA, self-amplifying core mRNA, which is not being effective by any mutation. then they are basically effective against all variants, right? So until the development of those vaccines, I think there will be a combinational therapy for at least a year, which means like you are using some sort of like the regular 2 doses of the vaccinations or 1 dose of the vaccination, whichever you are going to take, together with this pill. So because that would be actually, I think, more effective thing, I would say. But in addition, I would say one more thing actually I would like to add. Just give me 1 second. I think I have it here somewhere. The question that we all were going to take some boosters. They have told us to take the boosters, that was definitely because our overall protection against these vaccines, their efficacy actually, and also to enhance the herd immunity. And yeah, so I think the future would be our 2 vaccines, maybe the booster shot, but not everyone has to take the booster shot, but together with the Merck pill, I think that would be the most effective, because it is, if you see here, it's a very good strategy. It is already into the phase 3, I think, clinical trial. Merck's it is already approved by FDA, but for long term, I don't think so. I think until we get the next generation of vaccines, then I think we probably would not need to take these pills. But for now, maybe, because I read somewhere that these pills together with vaccination, it reduces the chances of death by 50% actually in COVID-19 affected patients.
Okay, thank you so much. Thank you so much, Noeen, for answering the question. I think we have another question from Makoha. She was just commenting on about maybe she wanted some more elaboration on that dosage intake, of which I think, yeah, maybe from the previous answers, I think she got something. Maybe some comments on that dosage intake.
Which index? This index? Which index? Sorry. Can you hear me? Which index? I didn't get you.
Hello.
Yeah, which index?
Hello.
Okay, Jim, you can go ahead.
Okay. Maybe, yeah. Someone is asking the Risi Makoha. Maybe she wanted you to comment more on that dosage intake. Maybe I'm not getting well the question, but maybe
She's talking about this intake.
I think intake of the vaccine, maybe. Yeah, I'm not getting well the question.
Okay, I can answer this. Like for intake of vaccination, anyway, vaccinations are very important for your safety. Definitely, there is no question about it. And the intake of the pill, I think it depends on it is mainly, as far as I get it, it's not required for people who are vaccinated. I mean, for example, I am fully vaccinated. I don't need to take this pill. I am required maybe in future with time, maybe after a few months, to get the booster shot, just to get the protection again, because this protection maybe is reducing inside my body, like in vivo. But I am not required to take this pill. I think this pill or this therapy is basically for COVID-19 patients who are already affected by it, so that they can test its efficacy in the combinational therapy. That's what I understand. I don't think that people who are vaccinated, they are required to take the pill. I don't think this is the case. If they are vaccinated, they are well, but they are definitely required to get vaccinated. Vaccinations are anyway far more effective and more efficient than the pills. Then anything that is like administered in you in the form of injection is more effective than the oral administration. It just requires more time to show its efficacy in the body.
Okay, thank you so much. Thank you so much, Dr. Noeen. Someone else is asking about, that's Ojambo Kevin. She is saying, thanks for the presentation. Or maybe I can begin on the other one. Maybe, yeah, from Daniel. Hi, Dr. Malik. Please expound more on the next generation vaccines. What is their predicted mechanism of action? I missed on that. Thank you.
Next generation of medicine vaccines. So first, next generation of vaccines. One, sorry, first thing is the trans self-amplifying RNA vaccines, which are addressing the many issues like immunogenicity of the self-amplifying mRNA-based vaccines. In addition, it is also um, addressing the interferon activation, which leads to the degradation of mRNA with time. So it also is addressing that issue. In addition, but those vaccination will take time to be more commercial. So in addition, I cannot disclose the information, but there is one company that is working on the vaccines, which is using, I think they are using the RNA from the core, not from the spike proteins, but from the core. So whenever mutation happens, the mutation basically changes the information in the spike proteins. So that's why many vaccinations fail, but I think they are still in the development process. So maybe that will also be the next generation, fall into the next generation category of the vaccines. Whenever it will finish, then definitely it will be more effective for all sorts of variants. And so that's my thing. And the other thing is also the storage, because if you see here, I think if you see here, I think most of these you see here, most of these vaccines, they require the storage, like the effective vaccines like RNA are most effective vaccines. They require the storage of -80 to -60, which is very crucial, for many countries. They don't have good storage system, because you definitely need a good system to store these vaccines, but also they are very effective, far more effective than the viral vector or inactivated based virus based vaccines. But here the storage conditions are desirable, which many countries can get or can have it. But this is very crucial point for Pfizer especially, and somehow for Moderna too, for many countries. So I think with time, the development of new vaccines, which is also taking into consideration the storage system to help the shelf value or stability of the mRNA based vaccines would be very important.
Thank you so much. Thank you so much, Doc. For the next question from Ojambo Kevin. Thanks for the presentation. Why is it that mRNA vaccines are associated with cardiovascular system damage? Then why is there a need for frequent rehydration after getting vaccinated with mRNA vaccines? Thank you, Doc.
Sorry, why it is need to get hydrated after the vaccines? This was the question.
Yeah, it's frequent rehydration after getting vaccinated with mRNA vaccines. Why is it so?
Okay. I got this one, Moderna. I did not go through this situation actually. For me, the only thing that I got was my little bit sore arm. But of course, you need to get like more and more hydrated for any type of the virus things. You need to be more hydrated. I am not like a physician, so I cannot answer this question on the medical perspectives, but I think it also depends from person to person, these conditions.
Okay,
I
so much,
cannot
Doc.
actually comment on the medical side, because that's not I'm not a physician for vaccines basically.
Okay, thank you so much. I think we don't have any other question. Maybe, yeah, Doc. I just wanted you to maybe, for example, as far as the mRNA vaccines are concerned, we know that they are very specific, highly specific. Now, due to maybe development of technology, in medicine, will it be a possibility of developing a multi-epitope vaccine, maybe by combination of different mRNAs to come up with a vaccine that is capable of inducing immunity against several various infections? Thank you.
Yeah, actually that is possible. I actually talked about it before also. You can always use multiple type of antigens into your mRNA constructs to address different issues. That's definitely possible, and that can also definitely be done. And I think there are some groups who are already working on it. So I think that would be more effective. Yes, that's definitely is one of the future perspectives.
Thank you so much. Maybe next, I just want maybe, for example, some elaboration maybe on the preparation of the working seed, the antigen isolation and purification.
Or what is the question of here the preparation of antigen, or like what was the question?
Oh, okay. Yeah, my question is for vaccines production, manufacturing and production, you have to have the working seed, now the antigen. Now, how do you go about by isolating from the source? And also you prepare the working seed now.
Yeah, so
Isolation and
for
purification.
purification and isolation is here that I actually talked about it before. Let me actually repeat it. Just give me one slide, 1 minute. Yeah, actually here is, if you see here, let me talk about it a little bit in detail. So for purification, we all know that mRNA has a negatively charged phosphodiester backbone, and many of the purification techniques which are used for DNA removal or DNA purification, usually in experiments, can be adapted for the purification of this molecule also, just to get rid of NTPs, enzymes, and DNA fragments which you need to be removed to get a pure mRNA. So these techniques involve the size exclusion. These are different types of chromatographies, like size exclusion chromatography. I think this is the most common. Many of you have used it. Reverse phase chromatography is also many of you have used. Anion exchange chromatography, hydrophobic interaction chromatography, thiophilic adsorption chromatography. These all chromatographic methods actually use different columns and use different detectors, right? So you can definitely use many of these techniques. And for routine preclinical work and in vivo immunization studies, RNA can simply be precipitated. Why it can be? Because it is already polar nature because of negatively charged side of mRNA, which makes it highly soluble in the water actually. And if you add several cations like lithium chloride in combination with ethanol, like ice cold, you can make it cold as a co-solvent, then you are actually neutralizing that negative end of the mRNA, and it helps then decreasing when you're neutralizing that negative, then of course it is no longer polar, and when it is no longer polar, its solubility is decreased in the solution. It can get precipitated. This is another method, but it is not literally more ideal method. So definitely there are some other methods which are used, and which these methods, for example ion exchange, affinity, and size exclusion for purification, are still in you can say reports in research. It is not, there are still some articles coming out. There is not one final thing, one final solution that you can use. But of course what you can use this purification method, and you can combine it together with tangential flow filtration method, and in this method actually you are actually good, it is very good for RNAs where they are in the size in the order of 10,000 bases or larger than 3 million Dalton. It is very important because using this, you are actually removing the lower molecular weight species, but you have to use the membrane with the right molecular weight to cut off all extra things. And at the end, you can always add it to the buffer solution, and you can just like adjust the pH to the required concentration, and then you can do the sterile filtration using the sterile filters. So I think the sterile filter size is in the, I forgot its size, the number that you can use. I think it was 0.2 micro or something like that. I just forgot the number for filter size that you can use.
Okay, thank you so much, Doc. Actually, a wonderful presentation, a very excellent one. Actually, we have learned a lot. Yeah, thank you so much for answering all the questions, Drew. Thank you so much for the presentation, Doc. As students against Superbugs Africa, this is an organization of the students. Actually, we have a number of young researchers, young upcoming researchers who are interested in joining maybe some of these research organizations. We are so much grateful for having you today, taking us through some of these advances in technology, the mRNA vaccines. I know many of the people that do not know much about the production, development, and also the effectiveness of the mRNA vaccines. But I'm quite sure that most of us here, we have got something to learn and to add on our knowledge. Thank you so much. Maybe I don't know if anyone has any other question before we proceed on to the closing remarks. Anyone?
I can actually now see that question clearly in the chat box. It was about why is that mRNA vaccine are associated with cardiovascular system damage. I think it varies from person to person. It's very rare. I think it's 0 point something or 1% of the whole population, or I don't know what are the exact numbers, but it is very rare. It again depends on the person to person. Majority of the people, they don't get it. Majority of the people, they don't even get that inflammation of the arm, and some get after getting mRNA vaccine, like the sore and heavy arm, like because it is intramuscular. And with any vaccination which is intramuscular, you always get the sore arm, whether you are getting the flu shot, because it is going into your muscles, and then of course your system, all your immune cells are rushing towards there, because there is a foreign particle there to fight that, and then it gets a little bit, makes your arm a little bit heavy. So this is like a simple and lay explanation for that. So this is all like most common thing that I can tell. Otherwise that's not very common.
Excuse me, before we... Hello?
Hello, yes, go on.
Hello?
Yeah, Manzama, yes, please.
Yeah, yes, thank you. Sorry, I have one more question. I would like to ask, is it possible to explain a little bit deeper about that mRNA engineering, like that's how to engineer that, the mRNA itself? Because in the slide I saw like the picture who shows some structures or something like that, but
Yeah,
it's not so clear.
it's okay. It's by the coding. Actually, you are doing the genetic coding, right? So you are encoding that transcript inside your mRNA construct. So what I can do is, let me share my screen again.
Okay.
I think this was my full presentation, right? Okay, so let me share my screen and go back. If this was the slide, right? So you see, this is the different DNA construct actually, which is encoding. You need to add engineer the coding basically in your vector design. And that's how it will be go. But in detail, if you want to read, you can always because most of this, like all of this lecture is based on these review articles, which are excellent review articles. You can
Okay.
always go to these papers and read even more detail. And
Okay.
if you cannot download these papers, these are, you see, these are very recent from 2021 and 2020. If you cannot download these papers, you can note down, I think, right now if you want. If you cannot download, then let me know. I have these PDF files and I can always send you by email. But this all lecture is based on these review article, and they're excellent to get any information.
All right. Thank
More
you.
detail, like more sequencing, like which sequence you want to use for the
Ah,
DNA, or which sequence has been reported, or which are still in process. So these papers are really good. It will always take you back to the other research articles.
all right. Maybe I can drop my email. Where can I leave my email so that I can have the PDF of these papers? Since they are very recent, it may
Yeah,
be
sure.
hard for me to get them.
I can actually drop my LinkedIn ID, so you can even connect with me either on LinkedIn, and I will also drop my email address. You can even connect with me on for everyone who wants to connect. I am dropping my LinkedIn ID in the chat box, and I'm also dropping my email address. So if you would like, you can write me email. Okay,
Okay, fine. Thank you.
no worries.
Thank you so much.
No.
Okay, thank you so much, Doc. Thank you, Doc, for taking us through this presentation. Actually, we have learned a lot. It was a very nice one. We are so much grateful for having you today here. And we are hopeful that you'll be you'll be willing to support us any other time that we need you. Actually, yeah, because we are going to have many other webinars ahead of us. We would like to invite you any other time that we will need you. Thank you so much. Maybe before we end the session, I'd like to invite Daniel to say something before we end. Dan.
Yeah, thank you so much, Jimmy. Thank you so much. Thank you so much, Dr. Malik, for the very insightful presentation. I was very intrigued, and I was really some of these things I never even knew, and it's a pleasure to have you today, and for you, to create some time from your busy schedule to share with us this knowledge pro bono. We really do not take it for granted. We appreciate so much. Gather also sent his appreciations. He had to leave a bit early because he had another meeting that he had to attend to. But he told me, he sent his regards. He's really happy that you agreed to attend to the session. And I would say, yeah, we'll have you once again. We hope that we'll invite you once more. And now for nuclear medicine, your area of expertise, I'm sure you'll also prepare something very good for us. Because it's even your area of expertise. As Jimmy said, we are working on a FENS program. The webinar series is just, the introduction, but next year from around January to around May, we are working on a FENS program where we'll have a select number of students, those ones who have been attending these webinars, undergo some intensive training and, have these sessions in a more intensive way and in a more practical approach. We also hope to have secure some placements for some of the students who will participate in the program, maybe in local pharma companies or research institutions, or even foreign ones, if we're able to get some scholarship arrangements. So we will contact you, we'll keep you in the loop, and so that at least can also help us in organizing this FENS program that we are currently developing. So once more, thank you so much. Thank you so much, everyone who attended the session. Thank you for making time, and we'll share the recording with you. We'll share it via YouTube, and please, you've been able to attend. Maybe there are some others who could not attend because of some other competing interests or commitments. Please share the link with them We also have a WhatsApp group where we share all this information. So if you're not in the WhatsApp group, you can contact me or Gadai, or Jimmy. Jimmy, you can just drop your email maybe in the chat box. For maybe anyone who is not in the chat, who is not in a WhatsApp group, you can join, so at least we're also able to engage in these discussions. And when times comes for us to have the defense program, at least you also in the know and you'll also be considered. So that's it. And thank you so much, everyone. Thank you once more. And thank you, Dr. Malik. Thank you, Jimmy, for us coordinating the session. And I wish us all a wonderful evening. Maybe Dr. Malik, if you're in Europe, a wonderful evening. If you're in the US, a wonderful day ahead. So thank you so much, everyone. And over to you, Jimmy. Thank you.
Thank you so much, Daniel. Thank you so much for the kind and wonderful closing remarks. Thank you, Dr. Malik. Thank you so much. We would like to invite you anymore, most easily for the defense program that is ahead of us, starting on January. Actually, we are preparing with some topics of nuclear medicine, and we would really like to have you back soon. Thank you so much, everyone who has made this day a success. Thank you, everyone who has attended this session. Actually, it matters a lot. We're so much grateful for your presence. Maybe there's more other activities that we would have engaged in, but actually, that's why we really appreciate you who are present today. Thank you so much. Maybe, for those who are not in the WhatsApp group, I can just share the link with you. Let me just share the link, in a bit.
Okay, thanks everyone. Thanks Daniel and Jim for invitation. And I think Stefan, he's not here. How you call him Gatai, not Stefan, right? I'm calling him Stefan. I don't know why. But thanks for the invitation. It was really nice being here. And I am happy that my presentation could bring some information to all of you. It was just I prepared in the form of a lecture, and anyone who would like to connect with me, I have dropped my LinkedIn and my email ID in chat box, and I would like to be in that WhatsApp group, so I am also dropping my phone number, so you can add me too in your chat box in the group. And I am actually based in Canada, so it is actually 7:30 a.m. in the morning time, so I still have a full day ahead. So I wish all of you a wonderful evening and wonderful late afternoon. I think it's already going to be evening soon. So, and a very nice and lovely weekend. Thanks for inviting me and having me here. Thank you all.
Thank you so much, Doc. We are so much grateful.
And for making time in the morning, I think we started around, it was maybe 6 a.m. or something, we don't take it for granted. Thank you so much for the passion.
It was 6 a.m., 5:45.
Okay, thank you so much. Thank you so much. Yeah. Over to you, Jimmy, to close us off.
Thank you, Daniel. Thank you again, Doc. Thank you, everyone who is in the meeting. See you in our next meeting on Saturday as we go on. Maybe, I'll like to adjust to maybe, for example, I'll just to remind you, maybe, when it come to these sessions, let's take them seriously. Actually, we are learning a lot from this session. Let's invite our friends who are not in this in the WhatsApp group. We can just let them get in. We can share the link to the WhatsApp group. We would like to invite everyone to be part of us, actually, to improve the situations in Africa and even beyond, especially in the production of vaccines, drug. As far as antimicrobial resistance is concerned, the core purpose of this group is to fight against antimicrobial resistance. Without taking no much time, let me just wish you all the best. Have a nice time. Bye.
Thank you, everyone. Bye.
Thank you, everyone. Bye-bye.
Thank you, everyone. Bye.
Bye-bye.